Um, so yeah, we can get started now. So yeah, my name's Dylan Cable. I'm pleased to introduce Dr. Fei Chen visiting us from the Broad Institute. So, formal biography, Dr. Fei Chen is currently an associate professor at Harvard in the Department of Stem Cell Regenerative Biology, and he's also a core faculty member at the Broad Institute. And his lab focuses on developing tools for single cell genomics with spatial and temporal resolutions. And these tools have been really influential in helping us to kind of integrate microscopy, genomics, synthetic biology, and to interrogate transcriptional programs at a cellular and tissue level. So Dr. Fei Chen obtained his PhD in biological engineering from MIT, and he was a Schmidt Fellow at the Broad Institute previously. He's been awarded many awards, including the NIH Director's Early Independence Award and the Allen Distinguished Investigator Award. So a personal note is that Faye was also my PhD advisor, and I've personally known Faye to be an excellent scientist and mentor. I would say that Faye is an innovator and also a tinkerer, and he enjoys finding creative solutions to biotechnology problems, and he inspires the same in those around him. So with that, I'm very excited to introduce Dr. Fei Chen, and I will give it away to you.
All right, thank you, Dylan. Yeah. Um, can you see my screen? Can you see my
Yes.
screen? Okay.
Yes.
Yeah. Okay, great. Thank you for the introduction. Yeah, it's a pleasure to visit you guys virtually, and also to see Dylan. Um, so I'm excited to tell you about some work that my group has been doing on I think mostly on measuring and modeling tissue organization. It says across space and time. I think I'll talk a lot about the temporal aspect of it today. Um, I put up um this uh this this book by Arthur C. Clarke. It's kind of funny. He was a science fiction writer. I'm a big fan of science fiction. But this is a series of essays he wrote that are more futurist from the 60s, including some things about electronic brains, which are infinitely wise and benevolent, which might be relevant in today's world. But it's not random. So it's I think this could have been an alternate title of the talk, but he famously had these three laws of science fiction, which will come up later in the talk. Okay, so this introduction might, given the context and the department you guys are all in, you guys clearly know that cells are really important in the context. This is a picture of an H&E of a cell. It's hard to really make out the details, but when we zoom out, you can see it in the context of the tissue, right? And this is a Hodgkin lymphoma tumor cell. You can see a large nucleus surrounded by cells of varying shapes and sizes, and this might be information might be important for a pathologist who's like trying to make a diagnosis or something. But that's not why I'm pointing it out. I guess I'm pointing it out as an analogy, just like how it's really difficult to tell what's going on in tissue by zooming in into like a cropped image of a single cell. Genomics has often been focused on dissociated single cells, and akin to like zooming into that microscope image there. And we were actually become really good at measuring the properties of single cells, their genomes, their transcriptomes, and epigenomes, and it's really taught us a lot. And I think if we were all single cell organisms, maybe we would be done. Um, but obviously in humans and multicellular organisms, the spatial context and tissues is critical. And so I don't have to explain that to you guys here. You guys are thinking about that all the time. But I also want to just say the spatial context is only part of the story because a cell's present state really also reflects how it got there. That includes like the lineage, the exposures, and the prior state transitions. For example, this lymphoma tissue was at some point probably in the past a norm, a lymph node, right? Which has its own tissue architecture. And the transition of how it evolved is maybe very informative for our understanding of the mechanisms of carcinogenesis, but also maybe how to treat it. And conventional genomics, therefore, has also really, we get to see the present, right? We get destructive snapshots. We don't get the history. And so a major challenge is then linking this present state with the past in the same biological sample. And so I think in today's talk, I'll give you a little bit of a an overview of the measurement technologies the lab has been developing. But most of the time, I'll spend on talking about how do we connect the present with the past using genomics. And so, some effort in my lab has been built trying to build like genomic measurements directly into tissues. This I feel like has a series of mature technologies now, spanning transcriptome, genomes, and epigenomes, as long with associated computational tools, for example, that really Dylan actually led, that are widely used. I could probably spend a whole talk on the technologies themselves, but I won't talk about the spatial technologies today, just a bit so we can talk about the temporal measurements. Um, but I just want to say that maybe for the purpose of the talk today, Arthur C. Clarke had one of these three laws. As I said, one of them is that sufficiently advanced technology is indistinguishable from magic. I bring that up because of two things. I think one is that these sorts of measurements, like being able to sequence the genome within a cell or being able to measure the gene expression of every single cell across a tissue, is like maybe would have been like magic, even a decade or two ago, right? And the second is that maybe that we don't have to focus on exactly how the technology works for this for some of the talk, rest of the talk, but what we can learn from the technologies. So I just want to highlight one aspect of how a lot of these technologies work, which is that a number of them build on the simple idea, which is these spatially barcoded bead arrays, where microbeads have these unique DNA barcodes and they're put on a slide. And we know the XY location of each one of these barcodes. And in the beginning, what we could do is we could, you know, these would allow us to capture DNA or RNA directly onto the slide. But more recently, we've developed a version of the technology, which we use for many things in the lab, and I'll introduce it because it'll be important for the rest of my talk. And it's called slide tags. And it kind of inverts these microbead arrays and does a different sort of thing, which is allows any single cell measurement to become a spatial measurement. And how that works is that um, the concept is quite straightforward. You can perform your single cell sequencing and know where your cells came from. And to achieve this, what we do is we put a piece of tissue onto one of these microbead arrays. And again, each one of these locations has a bead with a unique DNA barcode. And now these DNA is photocleavable. And so when you could after you put the tissue on, you can cleave these barcodes and diffuse into the nuclei of these cells, and they get attached basically non-specifically to the histones. And then when sequencing is performed, the barcodes reveal the spatial origin of each cell. It's kind of like GPS for the cells because each cell picks up a distribution of barcodes from nearby beads, and you can actually fit the centroid of that distribution to very high resolution, and then that allows single measurement cell measurements to include spatial information. And so the technology was published a couple years ago now, and so I won't go over exactly all the details, but I'll just say that a couple things. It allows any single cell workflow to move into the tissue context. Here, this is single cell 10x 3' on mouse hippocampus, and you can see the cells. Those are like actual single cell data, but then now you placed it into to space, and it's adaptable to genomic and epigenomic measurements, which you'll see is important later. And the other thing I would just say is that, because it's compatible with any single cell measurement technology, lately we've been really using it together with 10x Flex, which is an FFPE compatible single cell approach. And here is slide tags on some metastatic melanoma specimen where this is about 150,000 cells. And only 100,000 are shown here, but in total, there's about 150,000 cells. And you can see that you can get very high quality data. These are true single cell data with thousands of UMIs. This is a median of 6,000 UMIs per cell, a median of 3,000 genes per cell. And a directly adjacent section, we perform Visium HD, which is one of these like capture-based spatial technologies that's very similar to SlideSeq. And we also find this together with SlideSeq is that we get about a 10x higher molecular sensitivity. Using this sort of true single cell capture ability, and then if you just look at there's this other aspect which is that you can just look at the quality of the data by looking at the UMAP, and you can see that the true single cell nature gives you much higher separation between types of cells versus the mixture plus the low sampling just gives you kind of this ball in the Visium HD. All right, so um, you know this is why we really have swapped over to doing slide tags for a lot of things. And in general, we've been doing it entirely for almost all of the measurements in the lab. And um, so now I'll just transition to actually the main part of the talk, which is going to be three like different ways of measuring cellular history in tissues, most of which use the slide tag technology. And so if you just look at that piece of metastatic melanoma, right? You can see this is an H&E image. And H&E, for those of you who know, is usually like this like purple color, but you can see this dark, uh, brownish stain. That's not actually from the H&E, that's from the melanin in the tissue. The melanoma is like a cancer of melanocytes which produce melanin, right? And and oftentimes, actually, anecdotally, in this is in a sample that's metastatic and hasn't responded to therapy, as pathologists have noted that like when when there's lack of response, you can see this region of the tissue, for example, that has lost its melanin staining, right? It has basically there's this phenomenon where the tumor evolves and de-differentiates into a different, more mesenchymal-like state, and that generally makes it more resistant to drugs and more aggressive, right? And but you can see that there's even in the same region of the cancer, this is about a 5 millimeter section, you have regions that have lost its melanin or regions which still retain melanin. And also interstitially, in the previous plot, there were many immune cells. And so like, how did this evolve? But this single snapshot really doesn't tell us how the cells got there and what was their evolutionary history and how that, you know, interplays with other cells in the tissue. And so, if we want to understand the past trajectories that give rise to present tissue states, um, you know, we often don't have the luxury of sampling over time. So how can we look backwards in time from the measurements that we can make? And so in many ways, genomics can help us do this because different molecular layers reflect different time scales. So like, for example, the transcriptome might reflect something about the recent transcriptional identity or recent state. Although maybe there are ways to elongate what recent means here. The epigenome stores aspects of maybe past exposures and transcriptional identity, and the genome might really encode something about lineage, although it's actually in a form that might be difficult to read. And so I'll tell you in the next I guess 40 minutes how we've been looking at uh, building tools to look at each of these windows in tissues and in cells. And so um, I'll start with DNA, just working our way up. So DNA, is passed from cell to cell and could inherently record lineage because each time the DNA divides, there's a couple hundred, maybe 10 to 100 or more, depending on the cell type, single nucleotide variants. Um, but that's kind of like looking for like a needle in a haystack because there's 3 billion bases, right? But in cancer, the mutation rate is much higher. And oftentimes they have large chromosomal number alterations, which provide a useful handle. And so this made us think that maybe cancer is a good setting to start with to ask how DNA can be used for lineage in tissue context. And so we started this many years ago, where we first developed a way to use these spatially barcoded arrays to capture DNA directly and that allows us to read out copy number alterations in human tissues. And this is through the use of TN5 tagmentation, and then followed by using the array to capture the tagmented fragments. And when you do that, for example, here is a colorectal cancer specimen from a human. And you can see in the H&E here that there are these little aggregates of tumor cells surrounded by stroma. And here in the immunostaining, for example, you can see the tumor cells are expressing MKI67. These are dividing cells. And then in the stroma, you can see CD45 infiltrates and other cells. In the H&E, these cells like look kind of basically the same, right? Um, but when you read out the DNA, you can see the directly visualize the chromosomal number alterations these cells have. For example, almost all the tumor cells have chromosome 8 gain, 8q gain. It's a MYC-driven cancer, and MYC is on chromosome 8q. But then you can see some subtle differences in some cells, but not others. For example, this chromosome 20 gain, and and then when you do just simple clustering of these chromosome bins, you can assign subclonal assignments, which are basically means these cells are, even though they're very closely, this is a 3 millimeter diameter, they're spatially adjacent, but they've evolved over time to gain and lose different chromosomes, even though they're originated from a single parental clone. And this is the first time we were able to directly observe this spatial heterogeneity and clonal architecture in human samples. And what also struck us is this length scale at which this was occurring at, right? Often, obviously, people have done multi-region sampling of tumor to look at the heterogeneity, but this is the heterogeneity in clonal architecture at the scale of hundreds of microns to millimeters. Um, so that work was published, and we performed a lot of work trying to investigate kind of like the cell intrinsic versus cell extrinsic gene expression relationships between the tumor cells and their environment. But today I want to, and that was kind of a powerful framework, but we found that copy number alterations are generally pretty low resolution. And so we've been trying to get at some ways of getting finer scale lineage resolution. So in one way we've been doing that is we've been turning to evolving lineage barcodes, where Cas9 progressively edits a synthetic genomic locus as cells divide. So basically the Cas9 increases the mutation rate at a locus, and you can read it out by a DNA or RNA sequencing in this case, because the locus is also transcribed. And a couple of um, systems have been developed. We've been collaborating with Fernando Carmago's lab and Jonathan Weissman's lab in bringing these lineage barcoding systems into the tissue context. And I'll tell you about some collaborative work we have had with the Weissman lab using one example. So in this example, there is a mouse model that carries this evolving barcode called tracer. And it also has basically a KRAS P53 driven tumor system, where if you put in Cree sparsely, you can get single cells that seed into whole tumor clones. And then by reading out basically these tissues in SLY-seq or SLY-tags, actually, you can measure the transcriptome of these cells and then use the tracer to infer the lineage. And so this allows us to ask how lineage maps onto spatial context during tumor evolution. And we were particularly interested in basically in that question, but also this model allows us to look at both the primary as well as the metastases. So we can ask, are there particular niches that are important for tumor growth and for metastasis? And do the clones which seed the metastases sit in distinct niches in cellular neighborhoods? And so when we do this experiment, you can get data like this. This is a section of the lung. And we've colored in colors regions where we see these tracers, which mark the tumors, and group them by different seeding, like initial seeding cells. And so, for example, in this one region, you have a bunch of tumor cells which were seeded by one initial event. And here's the lineage tree from that seeding event. And here's just the tumor cells in that region. But you from this lineage tree, though, you can use the depth along these leaves to infer the subclones. And so, for example, here are 3 subclones highlighted here. And you can actually see the cool thing is that subclones are organized in space. They have some, they mostly are in spatially coherent regions, although, for example, you can see this large amount of dispersion in subclone one. And so this is kind of the general flavor of the data. And we performed this, there are many analyses you could do, but we performed this particularly, I thought, kind of interesting experiment where we sampled the primary tumor very extensively over many Z sections, so we could capture all of the primary clones. And then at the same time, we found a couple of metastases from in the same animal, like from the lymph nodes or the rib cage or the diaphragm, and we can measure those as well. And then because we have the lineage barcodes, we can map the metastatic clones back to their origin in the primary tumor. So what does that mean? So here is a single section, and you can see three regions. We've outlined three regions of tumor that has been seeded. And and here in this plot, this is the allelic distance to the metastasis. And so with these low allelic distances are cells which have very similar lineage barcodes to the Mets. And you can see that for this particular tumor clone, large tumor clone, you can see subclones within it, or subregions within it. We don't know if there's subclones yet, but I'll show you that they are subclones. Subregions within it that have really low allelic distance, suggesting this is where the Mets were seeded from. But you can see even closely adjacent regions where the Mets are not from. And what's different about these regions that the Mets are seeded from? Well, one is that if you just look at basically the rate of expansion in the lineage tree, you can see these regions that have low allelic distance, where the Mets are seeded, are also happen to be expanding. And the second is that if you look at gene expression of the neighborhood nearby cells, you can see that basically here is the seeding meta cells, which is this region of blue, um, and the Met have very similar immunosuppressive macrophage and hypoxic environments in the surrounding neighborhood. Versus actually, like if you look at other clones, which include basically this region and this region, um, even though they're from the same initial, like, the same tumor cells seeded these different clones, they have a very different microenvironment. And so, um, and and you can actually also confirm that in the histology, that these seeding regions are hypoxic and they have these suppressive microenvironments. And so what does that tell us? Well, one thing is that it suggests that maybe this seeding clone actually can reshape its surrounding microenvironment and then carry it into the metastasis, right? So there's some aspects of the microenvironment that are cell intrinsic to the tumor cell. Um, um, the sense that, you have a very similar microenvironment in the metastasis as to the seeding region. And then and so these metastases initiated lineages can share tissue contacts with the seeded metastasis. So this is lineage in tumors using these synthetic evolving barcodes. But what about in human tissues where you don't get to take advantage of kind of using this artificially high mutation rate? And where you don't get to take advantage of CNVs. So one thing we've been doing is we've been trying to do whole genome sequencing using slide tags. And in particular, we've been using this technology called the Resolvome, which is a commercialized version of PTA, which is this whole genome amplification technology. The main issue, not issue, but main feature is that takes place in plates. So it's lower throughput, but you get for each cell, you can get whole genome sequencing as well as the transcriptome. And so here, actually, we sorted from a single, this is a single CRC tumor, um, uh, 1,700 cells into plates, of which we sequenced 760 of them. And then in each one of these cells, we can measure the whole genome. And so we first sequenced at 0.1x whole genome sequencing. And you can also detect mRNA from these cells. And um, the the genome is actually quite well covered. Like the gene, you can see basically the Gini coefficient, which is how uniformly covered they are. These are really high quality whole genomes, although we haven't sequenced them that deeply. And then for each well, you can actually, for example, in 23M, which is like row 23, column M of a 384 well plate, this is the location of a cell. And so here you can place um, about 300, 600 of those cells. And then for 182 of these, actually, and then six normal cells, we then took some of those, we have all the libraries, so we can take them and sequence them deeper. And actually, we sequence them at 15x whole genome. Um, it's basically like the sequencing cost is the main rate limiting cost for this experiment. And so then 96% of those cells can be spatially mapped. Here you can see them in space. And from those, you can measure the single nucleotide variants. Jointly, there's about 13 million single nucleotide variants. Um, and I think on this plot, this is a heat map of the SNVs now clustered across these 182 cells. And you can kind of already appreciate that basically the columns are cells and then the rows are SNVs, that there are the two primary clades here and here. And then even within these primary clades, there are some subclades that are basically delineated by single nucleotide variants, right? And actually, you can look at these SNVs and they represent, for example, you can look at the mutational signatures. Some of them are some clock-like signatures. Others reflect, for example, mutational features of the tumor evolution, such as chemotherapy exposure. But the main thing is that from the SNVs, you can directly visualize these subclones in space without using CNVs at all. Right? There's about 20,000 SMBs that are shared, but you can see most of them are shared between all, like in the tumor initiating clone. But even here, there's thousands of single nucleotide variants that then delineate these two subclades, and then you can see the spatial heterogeneity of basically these individual subclonal events, right? At different node depths of the tree. And actually, you can see that, very reassuringly and very cool that basically like these subclonal events are increasingly spatially clustered, right? You're building up the tree in both space and in lineage. And you can measure basically like the spatial coherence of these little individual subclones and then the rate at which they're expanding. And so this is pretty early days on this whole genome sequencing side. Uh, but I think we can start inferring clonal growth dynamics, and as well as maybe mutational signatures that are correlated with these histological features. More interestingly, you can use this sort of approach, I think, to start performing lineage reconstructions in aged and healthy human tissues, not just cancer. Because most of the resolution in resolving these lineages is at like only a thousand SNVs between these subclades. So this is a very hard experiment, I think, to do, but is very exciting. Okay, so that's DNA. So if DNA records lineage, um, you know, the methylation can also record a different kind of history. It turns out that so there's many aspects of the epigenome. We've been looking at DNA methylation, it's some cellular memory of lineage because you can accumulate epimutations uh, in division and differentiation that are durable, as well as the regulation. And there's two aspects of it. One is that methylation at CPG islands, oftentimes at promoter gene start sites or in the first intron, regulates gene expression. It's a silencing mark. And then hypomethylation marks genes that are on in specific cell types. For example, CD19 is expressed in B cells, and you can see that it's highly methylated in all other cell types, but hypermethylated in B cells. Um, but more interestingly, I think also, if you cluster cells by their DNA methylation, this is a paper that other folks have done, you can see that the DNA methylation clustering reflects developmental trajectory versus if you cluster cells by transcriptome, you would see that it would reflect oftentimes basically transcriptomic state, right? Epithelial cells might cluster together versus here you can see that tissues of origin are grouped together. Okay, and so we've actually been developing some like single nucleotide, single nucleus, single cell methylation measurements, and then using that with slide tags. And so here is a single cell DNA methylation data set clustered on MCG now. Um, of the mouse hippocampus. And then we perform the standard slide tags measurements, and you can put this into like the spatial context. For example, here it's actually just a zoom in of the dentate gyrus and CA1, and you can see the granule cells as clustered by the methylation marks and the CA cells. And so what can you learn from this sort of data? Well, some things that this is like one of the first times you can measure single cell methylation in space at such high resolution, single cell resolution. And so some things you can just see by observing. We looked at the data, and one thing that neurons have that other cell types don't have is this accumulation of MCH, where MCH is methylation outside of CPG context. And interestingly, basically after birth, neurons accumulate MCH, and it accumulates MCH on coding genes. And again, it's like a repressive mark where it's reduced in genes that are expressed. But if you just look at the global MCH of these dentate granule cells, you can see just by eye that there's some sort of gradiented pattern where the gradient reflects kind of it's low here and high here on the outside. And we know from developmental biology that actually how this granule cell layer is constructed is that the inner granule cell layer is the cells are born and then they travel outwards. And so the cells on the very outside are the ones that were born in the embryonic time and then early postnatal and then adult. Actually, in the inside, there are some areas of adult neurogenesis. And it turns out then if you just fit this uh, from like the distance to the edge, sorry, this should be in microns, you can just see that the cells linearly accumulate basically MCH, probably as a function of their developmental age. It's, we don't quite know like if this is just a passive clock-like mechanism or if there's additional regulation for why this is happening. But this is something that no one has observed before, but we think that in the granule cells, this is what's happening. Um, if you look at if you go and try to discover spatial methylation patterns within cell types, in MCH, you will find that you can find spatially patterned methylation that's reflective of the underlying gene expression in RNA space. So, for example, um, high MCH corresponds to lower gene expression in that region. And so in CA neurons, you can see this gradient from CA3 to CA1, and you can again see high MCH here, and then lower MCH as the gene turns on. And similarly, you can see that gradient in the thalamic glutamatergic neuron here. So this is just two examples, but you can systematically discover this. When we do this sort of systematic discovery of spatially varying methylated regions, uh, here when we do it on MCG, which is because here we're looking at all cell types, not just neurons, an interesting thing we find is that many of these spatially variable methylated regions group by shared developmental origin. So what do I mean by that? What's happening here is that in this heat map, there are many, the columns are different spatially variable VMRs and the rows are cell types. And you can see that there are many that are shared by CA neurons, dentate granule neurons, and then this astrocyte population that says, and this is astrocyte telencephalon. And versus there's this non-telecephalic astrocyte population that has differential VMRs there. And you can visualize that for a single gene here, TCF4, it's a transcription factor that like in this region of the tissue, it's irrespective of cell type, granule cells. So here is just restricted for astrocytes. You can see that it's low in this region of the tissue and high in this region of the tissue. This is the telencephalic astrocytes. This is the non-telencephalic astrocytes. But you can see that that pattern is shared between the astrocytes and the neurons, which this is because, these astrocytes and the neurons have common progenitors and actually um, it's marking some shared developmental origin versus the oligonucleotides between NT and NT really don't have that pattern, and they don't have this shared progenitor system between at least these two regions. Okay. And so the other thing I would just say is that methylation in space was really previously unmeasurable, and measuring it really reveals new insights. One thing that, especially in MCH, we're observing many different aspects like this layering of granule cells. The other thing is that here's genome-wide MCH level. We know that really this thing in the brain, forebrain, really only turns on. This is from a 2020 paper. It really only turns on after P0. But we've been looking early, mid-embryonic. This is E16.5, and we observe basically patterning of MCH in specific regions in the posterior forebrain. And this patterning is in many genes related to neurogenesis and axonogenesis in the same way that it's regulated in adults. So it's, I think there's some aspect of regulation of using MCH that's turning on much earlier than what's been previously described. Okay, so that's methylation. And so now I'll talk about the transcriptome. And a common question that we often have is whether cells that made a particular decision of fate, for example, choice and differentiation, or maybe, when you treat a cell with a drug, when it lives or dies, it's a fate as well. Were they already different in the past before they made that choice? And we often imagine, right, trajectories while cells diverge in the past because of maybe subtle transcriptional differences or epigenomic differences, but it's very hard to answer, like, were they actually different in the past or is it stochastic? Given a fate in the future. And so for a long time, we wanted to make a time machine for cells. Can we go back and figure out what happened to what was different about a cell given its present choice? And so this first maybe sounds impossible. So this brings me to Clarke's second law. Really, the only way to discovering the limits of the possible is venture a little way past them to the impossible. And so what I mean by that is just that we spent a long time on this problem, even though it seemed kind of hard, and there were many things that didn't work. But we kind of were inspired by this idea of getting a cell to store its transcriptome, kind of like an elementary school time capsule. And so then you can later on and, ask once the decision has been made, what was different about them in the past. And we spent some time figuring out what was the right way to do that. And it turns out that um, we look for many basically protein capsule-like molecules, and it turns out there's already something in mammalian cells called the vault particle, which is quite fascinating that might be able to work as a time capsule. And I'll just introduce the vault particle now. They're these large hollow particles made out of 78 monomers of a single protein called the major vault protein. They were actually discovered by accident by electron microscopy in the 80s, and they're widely found in many mammalian cells. So we don't really know what their function is, but we actually wondered if they could be repurposed because of this large hollow nature as molecular time capsules. So that video was it opening up. You can see that it's basically hollow inside with this protein, uh, um, this vault PARP that's inside. And we reasoned that this large hollow structure could be repurposed for a place to store mRNA. And the trick was to take the int domain, which associates with the vault protein, and replace it with a poly A binding domain, and which allows it to capture mRNA. And the thought was that when these things assembled into the vault particle, the mRNA would be isolated and then stored to preserve the past transcriptome. And so um, we first wanted to know if such a thing it could, retain the transcriptome was stable. So what we did was we expressed what we call the time vault, which is just those two components, the major vault protein and the poly binding protein, and then in cells, and then we lysed them and added RNAs to them. And so if RNA was stored inside these vaults, then they could be protected, and then um, otherwise they would get degraded. And so here you can see just in qPCR a couple housekeeping genes that like only when you express basically the MVP protein, which is the capsule here, and the poly binding protein, do you get capture of or protection basically of the mRNA. So that means that it's captured, and everything else is degraded when you only have one or the other. And it's about a couple percentage of this is between 1 and 3% of the transcriptome. It turns out this capture is very reproducible. These are between two technical replicates. Bulk RNA-seq, extremely reproducible, and it's pretty high fidelity, the transcriptome. It's not perfect, and there are biases to the capture, but the Pearson correlation coefficient of 0.95. The stuff that there's some deviation that doesn't get captured by the vol, and those turn out to be things like mitochondrial genes or nuclear genes that cannot be captured because the vault is assembled in the cytoplasm. And we can overexpress these to be very abundant, and basically they're transcriptionally non-perturbative to cells. Here you can see that when we basically express the time vault system, the viability of cells is similar to if you just transfected GFP or PUC19. And when we do bulk RNA-seq, there's really no difference in gene expression induced by overexpression of these components, which is actually quite interesting. I think it suggests that these vault particles are extremely inert. And especially since, when you look at a microscopy image, there's thousands of these particles per cell. Here's counting, we counted over many cells that are transfected, and there's like on average like a 10 to 20,000 particles per cell. They're very well expressed. Okay. And importantly, they inherited stochastically into daughter cells, which means that you can pass down the recording in time. And so then one key question is how long are these recorded RNA stable? When we do this RNA protection assay, actually they can protect RNA cells, the RNA for like 2 weeks sitting in RNase. What about the stability inside of cells? So how do we actually do this recording? If you think about the time capsule analogy, you have to bury the thing at some time in the past, right? And so how we do that is we use an inducible promoter where we add docks and it expresses the vault, it stores the transcriptome, and then we stop it. And so then no longer is storing. And so we know it came from a particular window in the past. When we do that with 4SU pulse tracing, this is um, it's a 4SU experiment that allows you to label the transcriptome and ask how you can use that to infer the half-life of RNA that were made in the past. And when you look at the vaults, captured mRNA that was capturing RNA that was metabolically labeled, you can see that RNA has a half-life about a week, while non-captured RNA is like less than a day. And so that shows that basically it shows that like the capsules don't break up inside of cells and they don't exchange after being formed, and we can use it to really record RNA in the past. And so we then use this do experimental recording in a couple of different contexts. I'll just tell you about two. So in heat shock, we measured the heat shock response, which is really a positive control. And then we also measure this drug persistence in tumor persister cells. And so in heat shock, what happens is that we perform oh yeah. So how we do the experiment is again, use docks to open a window. And so here we did that from an experiment where for one day we recorded the transcriptome either in cells that we heat shocked or in cells that we did nothing to. Then we waited. And then in one of the groups, we heat shocked it again in the present, just right before we lysed the cells. And in other groups, we just did nothing. And so if you look at the recording, which is what is inside the vaults, you can see that heat shock genes are upregulated when we recorded it a condition where we heat shocked in the past, and nothing really happens in the recording when we do the heat shock in the present. That tells you that there's no exchange between these two compartments, and then exactly the opposite is in the present transcriptome, right? Because the genes for the heat shock have come back down. And this is the case for gene ontology. Basically, all of the genes that are differentially expressed are heat shock genes, and the recorded RNA only in the past, and then similarly with the present. Okay, so I think the system basically works pretty well to record past transcriptome states. And so one particular application we wanted to tackle with this technology was this idea that happens in cancer drug resistance. Cancer resistance to therapy is quite common and is a major driver of cancer mortality. And it's particularly relevant for targeted therapy. So today, what we can do is we can sequence a mutation you have. For example, you might have an EGFR mutation, and we can give you a targeted EGFR inhibitor. And what happens is that most people will respond to the treatment, but then after a while, they'll develop resistance, and almost all folks on targeted therapies acquire a resistance. And we know a lot about these resistance mechanisms at the state of acquired resistance. There are genetic escape mechanisms, such as just evolving to not be dependent on the target anymore. But there's this hypothesis and some observations that the seeds of this resistance begin earlier in these rare pre-existing drug tolerant cells that are present before therapy. These are not they don't necessarily have resistance mutations, but they're robust. They're like transcriptomically robust to being killed. Um, but it's hard to measure them, right? Because you need to understand what was different about them before you hit them with the drug. Um, so so and it's importantly it's very different than trying to understand the post-treatment response, because that's an adaptive response. You want to know were there pre-existing transcriptomic differences that made them robust. And so this phenomenon is you can observe it in persister cells. So this is PC9 cells, a small cell it's like a small non-small cell lung cancer cell line, and it has a mutation for EGFR that's targetable by this osimertinib, which is a EGFR inhibitor. And you can see when you do that, about 20% of the cells survive. And then when you grow out the cells again and treat them with the drug again 20% of the survive. So you're not selecting for a genetic resistant population, but a non-genetic resistant cell population. And so what we did was using the PC9 lung cancer cell line, what we did was we recorded the transcriptomes of all cells in the naive state and sequenced, and then we treated with the drug. And then in the cells that lived, we sequenced what was different about their transcriptomes in the recording compared to cells that were untreated, but also recorded. And this is last us asked what was different about those cells before they had ever seen the drug. And what we found is, for example, that cells destined to survive had elevated oxidative phosphorylation and reduced expression of mitotic genes. This suggests kind of a more quiescent and a metabolically distinct state. So basically, the untreated cells are more replicating, and then they also depend more on glycolysis. And this is, this in general, people have known, in EMT or that more or cancer, these quiescent cancer cells are more robust to killing. Beyond metabolic shifts, you can identify a couple of programs associated with cells that go on to persist, including some metabolic enzymes, the steroid hormone and detoxification of reactive oxidation species. This is probably some way to be more, and it can detoxify the cell, alternative signaling, and then also ECM and adhesion programs. And so importantly, you can see that these are the top genes that are differentially expressed in the persisters before in the recording in the cells that would go on to be persist. Almost all of these genes, except for except for fibronectin, after treatment, if you just sequence the transcriptome, they no longer look like they're different, or maybe they have opposite relationships. And that's because when you hit them with the drug, they have this large transcriptomic change, and then it can obscure the pre-existing persister state. And so it turns out that if you just perform single cell RNA sequencing on drug naive cells, this is just the heterogeneity of cells in that never saw any drug. And if you just score them for these persister genes, like fibronectin 1 or PI3 or LCN2, you can see that they mark a little bit of a subpopulation that's high in all of these genes. And in particular, so that's just that maybe there's some sub-state, transcriptomic state that is already exists in the pre-existing population. And in particular, if you look at fibronectin, you can, in staining here, you can see that before you treat it with any drug, there's some rare cells that have fibronectin positive. But then when you treat with the drug, you can see it selects for that actually. There's about 50% of the cells are fibronectin high, maybe a little bit more. And um, then what we did was that we went and had tested the functional importance of many of these genes. Here, I'll just show it for fibronectin. What we did was we used siRNA to knock down fibronectin. And when you just do that on these PC9 cells, it really doesn't really have a effect on viability. If anything, the cells grow a little bit faster. But when you treat it with the drug, this is the 20% that always live. But when you treat with both the siRNA and the drug, you can see that it synergizes quite strongly. Actually, 70% of the cells that would have lived, now die, right? And actually this selects maybe against, these, you can see in this previous image that only a small, like 70 or yeah, not all of the cells are fibronectin positive. It's very possible that there's another population of cells that have a differing transcriptional state or mechanism of overcoming the resistance. But certainly fibronectin 1 knockdown synergizes with osimertinib to deplete these persister cells. And beyond fibronectin, we find two other pathways that could be modulated, although not as strongly, to perturb these persisters. And so, we think that this is a really powerful way to systematically probe drug persistence and potentially resistance. and causality. And so, um, just to summarize the talk on this section, just, the present tissue state reflects the history and local evolution. And in cancer, this lineage is coupled to the extracellular environment. In particular, maybe Mets can carry along the extracellular environment of the primary that seeded it. And, there are these different molecular types, and they can reveal distinct windows into the past. And one thing that I think is quite interesting is that why should we look at the past? Well, differences in past states are actually perturbations. They're natural perturbations that can allow us to separate cause and from effect, right? That's how we in the natural world observe causality. We look at what happens first, followed by what happens next, right? So if we could do that in tissues, that would allow us to study this. And so I'll just conclude with in the past, classical genomic measurements uh, like are don't have spatial or temporal context. I think right now we've built a lot of the foundational tools to allow us to do this. But these days we're quite excited about how to we predict and control tissue circuits in vivo, and that's something that I didn't get a chance to talk about, but it's something we're trying to work on in the lab. And so I'll just thank the people who did this work as well as the collaborators. There's many folks, and including alumni like Dylan, who's here, and thank you.
All right, thank you so much, Fei. Let's all thank Fei for the excellent, exciting talk. So we have some a little bit of time for some questions now. So let's open the floor to questions for Fei. Yes, Kong.
Hi, Faye. It's nice to see you here virtually. Good to hear our talk. It's like so exciting. And I really appreciate that, like you're really trying to record what happened in the past for gene expression. But at the same time, for the first part, like spatial, I think technically the spatial environment can also change over
Yeah.
time. And at the time that we measure it, it's still potentially like the metastasis like could potentially be different from at the exact time point when it was seeding. I wonder whether you like are working on some of the like directions of like whether it's possible to direct to kind of like actually record the spatial environment and that in the past or
That's a
it
good
could be
question. We tried, I mean, I think in synthetic systems, you can maybe do that. I mean, I think there were some folks in the lab who were interested, and I think other people who have worked on that, like these sort of like lipstick-like measurements, right? That uh um so I think in synthetic systems, I think that's certainly an area that we could do. We haven't had too much success, or we worked on a little bit. We haven't had too much success in it. It's much harder to obviously do in um, human or like in systems you don't have genetic access to, and so in many ways we can only infer it from something like lineage or methylation
right.
exposure or something, and so that's something we're thinking about, but you can't infer the microenvironment, so is what your point is, and I totally agree with that. I don't know how you can get around that.
Right. I just like this. I don't know how to design all the experiment. Just like curious whether you have like a guess of direction on that direction. But yeah, thank you.
Yeah.
Yeah, yeah, thank you, Kong. Next, we have a question from Toshiro.
Hi, nice to see you. This is a
Hi.
great talk, very exciting. I have two questions. One question is about the tracer experiment. And then you see tracer experiment,
Yeah.
CRISPR-based lineage tracer
Yeah.
experiment in the space. In the experiment, you observe the clones in the primary tumor, in the lung cancer.
Yeah.
You also found in the metastasis, and then you see similar microenvironment. I just wonder if you observe the transcription state of the such clones are also same or they are different.
Yeah, transcriptional states are similar, more similar than the non-seeding clones. Although I think I didn't mention, obviously once it's seeded in the metastasis, it continues to evolve. So they end up acquiring a bunch of like
Yeah.
fibrosis-like signatures. They like produce a lot more collagen. But in many ways, the transcriptome state, yeah, is more similar than non-seeding clones. And actually, I think Jonathan's group previously in a single cell paper showed that, and we recapitulated that, I think.
Yeah, I think that is a really cool. Cool. And then second question is about the board experiment.
Yeah.
And that is really cool and very exciting too. So if I understand correctly, that basically you transfect the cells with this component within the cells, and they reconstitute kind of the organelle-like structure
Yes.
in capsule.
Correct.
So I just wonder if you manipulate the localization of this complex, and then can you capture different type of the a messenger RNAs, or just rely on obey in front of the translation machinery?
Yeah, probably you could, although we don't know to what extent you can. Um, certainly we are already observing that in the sense that we have like strong bias against nucleus,
Mhm.
nuclear transcripts. Like we don't capture link RNAs very well. Um, and so if you could move it into specific subcellular compartments, like um, I guess maybe like neurons have subcellular, but like polarized cells, you could probably bias it towards different subcellular transcriptomes.
Yeah, that is really interesting. Yeah, thank you so much.
Thank you.
All right. Well, thanks everyone. I think we're at time. So let's thank Faye again for joining us today and really enjoyed the talk. Thank you, Faye.
Thank you. Bye.
All right. Goodbye.